Hello, my name is Colin Gardner, and I'm here to present my poster about the key role of focal adhesion kinase in idiopathic pulmonary fibrosis. Idiopathic pulmonary fibrosis, or IPF, is a chronic scarring lung disease of unknown cause, and it is a progressive disease with short survival. Median survival is only around 4 years, and the current medical therapies are suboptimal. There are currently two FDA-approved drugs for IPF, and both these drugs only slow the progression. They do not cure or completely treat the disease. And one of these drugs inhibits a 3 receptor tyrosine kinases, and so we decided to look at FAK, which is a kinase, focal adhesion kinase, a kinase expressed in fibroblasts, encoded by the gene code PTK2, and this gene has also been implicated with a lot of progressions in cancer, including cancer stemness, epithelial to mesenchymal transition, and fibrosis. And so we are looking at this gene, and we aim to characterize the expression level of FAK in IPF lung cells, specifically lung fibroblast cell lines and PBMCs. We will study FAK inhibition in vitro as a potential pathway for drug discovery. And we use publicly available NIH GEO databases to analyze gene expression, and then we confirm cell-specific expression through single-cell sequencing, and then in vitro, we validated an FAK inhibitor with fibrotic markers. And then so here is looking at a Kaplan-Meier survival curve showing the survival probability in IPF patients, specifically looking at the PBMC cell line PTK2 or focal adhesion kinase expression. And here we see a statistically significant result that the higher PTK2 expression is correlated with a higher chance of survival. The lung tissue sample databases did not include survival data, so just looking at the relative gene expression, we can see here on the left that there is a statistically significant difference between healthy control and IPF cells. The IPF had much higher PTK2 or FAK expression, and then the study on the right, a different cohort, showed that there was a difference between healthy control and early IPF patients and healthy control and advanced IPF patients. And the advanced was a lot more statistically significant compared to just the early IPF. And then to confirm those, we looked at single cell sequencing, and you can see here that the fibroblast cells specifically do have higher PTK2 expression. And interesting, you can see here in this green box that there is a unique subtype of fibroblasts found in IPF cells called the HAS1 high fibroblasts that express a lot more PTK2 than normal fibroblasts. And then here's our validation. We used defactinib, which is a commercially available FAK inhibitor, to reduce TGF-beta activated fibrotic markers. Here you can see in panel A, the Western blot, and then B is measuring the FAK, and then C specifically is measuring alpha smooth muscle actin, or alpha SMA, which is a fibrotic marker in IMR90 cells, or mature lung fibroblast cells. And so our data demonstrates that higher levels of PBMC PTK2 expression correlates with improved survival, and that FAK PTK2 expression was consistently elevated in lung tissue samples from IPF, and this suggests that there may be a compartmentalized response, and this was also confirmed by single-cell analysis, and defactinib reduced fibrotic markers. So further analysis of this gene, the FAK PTK2 gene, is necessary, in order to develop new therapeutic options and IPF biomarkers to detect, prevent, and treat IPF. Thank you so much.